And the microscope, why it is required? So normally for the biological activities and all, some of the magnifications are required in order to find out some the internal parameters. Okay, and there will be a resolution we can able to find out. Even we can able to find out the distance between the nanoparticles also. Please look here. The main idea of using this microscope to study this is it is also mechanical properties we can say, but the mechanical properties will be studying the yield stress, okay, and thermal stresses, so many other parameters will be studying. But once after synthesizing the nanoparticles, those will be arranged in a systematic pattern, and now the microscopes will be used to find out the distance between these nanoparticles and any other things which are emitted or dissolved along with them to increase or to enhance the mechanical properties. All those can be studied. Okay, and one can see that if the microscope is having very poor nature, the magnification, what happens? There will be any structure will be formed. That means two bubbled blurred surfaces we can able to visualize, but there will not be much more clarity. But another thing you can see that the two dots and there will be a distance between them, and any other the particles or the composite material we are going to prepare along with these nanoparticles, we can able to study them clearly by using the microscope. Okay, microscope, electron beam microscope, under them the transmitted electron microscope or scanning microscope. Okay, like that many other microscopes are available, and we'll try to study them up one by one now. The optical microscopy. So optical microscopy, it is a technique employed to view closely the magnified the image of the object through the magnification lens. Okay, these optical microscopes sometimes they are also called as light microscope because of we are going to have the series of lenses to magnify them and through visible light, so that they are called optical microscopes or light microscopes also. And there may be the actually here what happens, one end there will be viewer, okay, and other end there will be this one lens will be other end, and in between them a sample will be kept. You understood my point? The lens will be placed in between the sample and the viewer. Okay, the sample and that means the sample will be kept first, then the next lens, then next to the viewers. So this is how the optical microscope will be arranged. And the simple type of these simple microscope, compound microscope, digital microscope, stereo microscope, and comparison microscope and inverted microscope. All these are meant for some kind of specific applications they'll be using. Okay, and in nanoparticles you can see that gel sol the nanoparticles or nanoparticles which will be suspended in the solids, suspended in the gel, suspended in the liquid, suspended in the air. For all of them we can have the specific type of microscope among them. Okay, now what are the applications and limitations of these? The optical microscope, we can ensure that the microscope will be set correctly. We should have to ensure ourselves the microscope is to get the output effectively from the microscope. One has to have to ensure there will be a correct setting up of microscope, just like your vernier calipers and the screw calipers. What happens, the accuracy will be based on how you are going to do the measurement in the similar way. And the object lens is brought closer and closer to the sample. Through a proper lighting technique, we can able to magnify them. Okay, we can able to magnify them. And to have a enlarged image of that, then the inverted image of the sample can be viewed through the eyepiece of the microscope. Okay, that means the inverted image in the sense it is like this. You can see here. You can see the object is very simple, smaller in one, and there will be a telescopic lens, And by moving the telescopic lens in a proper distance, what happens? We can able to view the magnified image of the object in the inverted form in these things. Okay. And now the optical microscope is commonly used in many research activities like microbiology, microelectronics, and nanophysics. Okay, biotechnology, pharmaculture activities. Okay, for all these will be used to view the inner sense of the systems in a micro level. Okay, and the optical microscopes are not suitable for some of the systems. Some of the systems that means they will be limited for some of the techniques. For example, if you see, if you are going to do the very high magnification of certain things, what happens? As I told you, the airy disk will be visible. You can see that. In some other applications, it may not be applicable because what happens now, when you are going to do the magnification to a higher extent to view the inner sight of the or inner information of the object, what happens? Instead of we get the clarity of the object, there will be airy bubble structure will be formed, and it is not possible to look into this. Okay, and to examine it by the viewers, it will become very difficult. Okay, it will become very difficult. In such cases, we are going to have limitation of this, and we have to yield towards other methods. That is called the scanning electron microscope. The scanning electron microscope, it is also called as SEM, and another one is there TEM, that is transmitted electron microscope. Scanning electron microscope, transmitted electron microscope, or simply we can say SEM and TEM. SEM, you can see that the scanning, as the name indicates, it will be scan the object. And another one, the transmitted electron microscope, it will be transmitted through the specimen. It will be transmitted through the specimen. And in both the cases, there will be electrons. The electrons will be, the electron beam will be the heated up in the anode. From the anode, it will be liberated. At a threshold temperature, what happens? The electrons will be emitted from the barrier, and it will be attracted towards the anode. And while it is moving in such a way that what happens? It will be scanning in case of scanning electron microscope. In case of the transmitted electron microscope, what happens? The electrons will be transmitted through this, and the scattered the scattered electrons after reflection will be rearranged in such a way that it will be showing you the magnified image of the object. This is how the working principle of both the SEM and the TEM. Those we can see them later now here. Okay, in this SEM, scanning electron microscope, please do not get confused with a scanning electron microscope and the transmitted electron microscope. Do not get confused. Okay, you should not get confused with them. And you can see here in the scanning electron microscope, the specimen will be kept here. And the transmitted electron microscope, somewhere the specimen will be kept here. You can see this is a sample. The electron run from the cathode at threshold temperature. The electrons will be liberated, and they will be moving towards this sample. And you can see that the the condenser lens, the primary condenser lens and secondary condenser lens, and the final lens aperture and the deflection coils, all these will be arranged in such a way that so that the electrons which are liberated from the electron gun should not be scattered here, that they should be focused and directed towards the specimen only, so that there will not be loss of the electrons. At the same time, to have the effective output, the electrons which are liberated from this electron gun should be attracted towards the specimen, and so that the more and more space is covered with the electrons, the more and the more information we can get from the specimen. That is the objective of this. Okay, and once the electrons are attracted by the sample, those will be detected by the secondary electron detector, and all this will be happened within this vacuum system only. That means it will be evacuated system. There will not be air because the electrons will not move properly in the air. It should be vacuum. A vacuum chamber will be there. In this vacuum chamber, those will be there. Okay, and the X-ray detector will be there. The X-ray detector, what happens so that the electron beam microscope should not be kept in the area wherein lot of the dense population is there. We are not going to keep it. Okay, the scanning, because there is a possibility that the X-rays will be leaked out and it will be entered into the human body and it will be dangerous. Okay, it will be having certain limitations. One has to take much more care about this electron beam microscope. Okay, the